In this video we will discuss about N-cadherin and MMP9 interplay. So idea is that this N-cadherins, even though it is overexpressed in the mesochemical type cells, but sometimes N-cadherins are not available on the cell surface. And one of the reasons could be that this MMP9 is also increased in some of the cancer cells in the mesochemical conditions. It might cleave off this N-cadherin. So they wanted to check whether there is any role of this N-cadherin and MMP9 or whether they interact with each other or not. So they in order to check this, they used the condition media, that is monocyte derived condition media and the macrophage derived condition media on two different cells. This NPC039, NPC076. And first they check the cell invasions, and they are able to see that this macrophage derived condition media gives more cell invasions for both the cases compared to the monocyte derived condition media. And these things they have validated using the quantifications also, that is shown on the bottom side of this panel A, where we can see that macrophage derived condition media has significantly higher cell invasions compared to the monocyte derived condition media. Afterwards they move to the main things, that is they check the N-cadherins, E-cadherins and MMP9 levels, since both of them, this N-cadherin and E-cadherins, plays an important role whenever the cells transit from epithelial to mesochemical. That is, E-cadherin usually goes down and N-cadherin goes up. However, in this experiment they are not doing this EMT transition, they are just checking whether there is a impact of the N-cadherin levels in the presence of this condition media, monocyte derived condition media and macrophage derived condition media. And they are able to see that when they check it for the monocyte derived condition media, there is no MMP9 expressions, and there is no impact on this N-cadherin and E-cadherin levels also, and this is the control KFDH. Afterwards, when they check it for the monocyte derived condition media, they are able to see compared to the in the macrophage derived condition media, they are able to see compared to the monocyte derived condition media that there is a decrease in the level of N-cadherins, but there is no change in the E-cadherin. However, they see MMP9 overexpressions. In the monocyte derived condition media, there is no MMP9. So this could indicate that this macrophage derived condition media somehow cause these MMP9 expressions because of the growth factor, or their condition media already contain MMP9. That thing is not established till now from these experiments. We will discuss whether they have did these studies in this future experiment or in the further experiment of this paper or not. But so far, let's focus on the results that are presented in this figure. So they are able to see in the macrophage derived condition media, the N-cadherin levels goes down, but there is no effect on E-cadherin, and the MMP9 cadherin level goes up. And they did this study 2 times, and both the times they are able to see the similar things. That is, in the case of macrophage derived condition media, the N-cadherin level goes down, no impact on the E-cadherin, and this MMP9 levels also goes up. Afterwards, since there are 2 changes that they are able to see, MMP9 and goes up and N-cadherin goes down. Now they wanted to check whether MMP9 and N-cadherin are as a role or not. In order to check that, they used MMP9 inhibitors, that is gamma L. And what they are able to see, for the control they do not use any macrophages or macrophage derived condition media or gamma L inhibitors. In that case, they do not see any MMP9. And when they use this macrophage derived condition media, they are able to see MMP9 and slightly decrease in the N-cadherin levels. And however, when they use these gamma L inhibitors also, in that case they are able to see a decrease in the MMP9 levels, and these N-cadherin levels seems to be similar to of that found in the case where they used macrophage derived condition media. However, they see another construct, that is of 40 kilodalton. So the idea is that this MMP9, whatever the MMP9 are presents, they get cleaved off the full N-cadherin levels. But when the MMP9 level goes down, this cleaving is become inefficient so far. That's why they are able to see these 40 kilodaltons also N-cadherin levels. So this is what, and also this MMP9 goes down is corresponding to this 40 kilodalton signals for the N-cadherin. So this all in together indicates that this macrophage derived conditioned media has a role towards cleaving of this N-cadherin levels, and as its level goes down, we can see the cleavage part also, and it can be expected that if MMP9 level completely goes down, this cleavage will also reduce, and this might show up increase in the signal for the N-cadherin so far based on these results. So I hope you understood how they made an interplay between N-cadherin and MMP9, and we will discuss further results in our subsequent videos. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.